Imagine a world where defeating cancer is not a matter of merely slowing a relentless enemy, but of surgically removing its engine entirely. What if, instead of constantly fighting to suppress the disease, medicine could erase cancer's key drivers the way a mechanic swaps out a faulty cog? This is not just the stuff of science fiction anymore. Right now, in the dynamic and high-stakes world of biotechnology, a bold new vision is taking shape, one that has the potential to fundamentally transform cancer treatment as we know it. At the forefront of this revolution are two industry giants, Gilead Sciences, a company with a storied legacy in antiviral therapies and a growing footprint in oncology, and Kymera Therapeutics, a rising star in the field of targeted protein degradation. These two powerhouses have forged a strategic partnership to develop a groundbreaking class of cancer drugs known as molecular glue degraders, or MGDs. This alliance is not just a business deal. It is a bet on a paradigm shift in how humanity confronts the complexities of cancer. But what makes this partnership so electrifying for scientists, investors, and patients alike? To grasp the full significance, we need to journey back through the history of cancer treatment. For decades, most cancer drugs have worked by inhibiting or blocking the function of rogue proteins that drive unchecked cell growth. Picture these drugs as highway patrol officers setting up roadblocks, attempting to slow the dangerous traffic of cancer cells. While this approach has led to important advances, think of the emergence of targeted therapies for breast, lung, and blood cancers. It is far from a perfect solution. Cancer cells are notoriously adaptable. Like clever drivers, they find detours, develop resistance, and continue their relentless march. The result is that many cancers eventually outmaneuver our best medicines, leaving patients and doctors with limited options. The persistent problem of drug resistance has driven researchers to explore entirely new ways of disarming cancer. Enter the concept of molecular glue degraders. Unlike traditional inhibitors, which act as temporary stoppers, MGDs function more like precision-guided magnets. They do not just impede the rogue protein, they tag it for complete removal. The cell's own waste disposal system then kicks in, breaking down and eliminating the troublesome molecule altogether. It's as if, instead of blocking a dangerous driver at a checkpoint, we remove their car from the road entirely. The main villain in this particular story is cyclin-dependent kinase 2, or CDK2, a protein that plays a central role in the cell cycle. In healthy cells, CDK2 helps regulate when and how cells divide. But in many aggressive cancers, especially certain subtypes of breast cancer, CDK2 becomes overactive, acting as a relentless accelerator pedal for tumor growth. Previous attempts to inhibit CDK2 with traditional drugs have stumbled, largely because these inhibitors often affect other, similar proteins, leading to toxicity and side effects. Moreover, completely blocking CDK2 without distinction can harm normal cells, causing collateral damage. This is where Kymera's innovation shines. Leveraging advancements in artificial intelligence, machine learning, and next-generation protein structure modeling, Kymera's scientists have engineered MGDs that do not simply inhibit CDK2, they remove it from the cellular environment altogether. These molecules are designed to be taken orally, a major advantage over many existing cancer therapies that require intravenous infusions or hospital visits. For patients, this promises not only greater convenience but potentially fewer side effects and better quality of life. In preclinical studies, Kymera's MGDs have demonstrated remarkable potency. Even at picomolar concentrations, a trillionth of a mole, these drugs have induced cell cycle arrest in laboratory models of cancer cells that are addicted to CDK2. This level of activity suggests a degree of selectivity and effectiveness that has eluded previous generations of drugs. According to Dr. Nello Manolfi, founder and CEO of Kymera Therapeutics, our highly specific, orally active CDK2 molecular glue degraders have shown a compelling preclinical profile and have the potential to transform the therapeutic landscape for breast cancer patients and other tumor types with high unmet medical need. For Gilead Sciences, a company best known for its blockbuster antiviral therapies, such as those for HIV and hepatitis C, this partnership marks a calculated expansion into next-generation oncology. Dr. Flavius Martin, Executive Vice President of Research at Gilead, explains, MGDs are opening exciting new possibilities in cancer research by offering a way to eliminate disease-driving proteins rather than just blocking them. This approach aligns perfectly with our framework of selectively targeting and killing cancer cells. But as with any bold new technology, the path from laboratory breakthrough to clinical reality is fraught with challenges and uncertainties. developing a drug that erases a protein, rather than merely blocking it, introduces new layers of complexity. The body's protein networks are intricately balanced, and removing a key player like CDK2 could have unforeseen ripple effects. Dr. Jane Smith at the Cancer Research Institute highlights this risk, cautioning, CDK2 has been a challenging target. Its role in normal cell function is significant. MGDs may offer a new avenue for therapeutic intervention, but only if selectivity and toxicity are meticulously managed. The stakes, both scientific and financial, are enormous. Under the terms of the agreement, Kymera could receive up to $750 million if all development and commercial milestones are achieved, plus tiered royalties should the drug reach the market. This is not just a windfall for a single company. It reflects the broader enthusiasm for molecular glue technology. Analysts project that the global market for these drugs could reach $5 billion by the year 2033, a testament to the immense demand for safer, more effective cancer treatments. Yet behind the buzz and billion-dollar projections lies a fundamental question. Can MGDs truly differentiate between cancerous and healthy cells? The crux of the matter is selectivity. If these drugs indiscriminately degrade CDK2 in all cells, the risk of side effects could outweigh the benefits. On the other hand, if Kymera's AI-driven design delivers on its promise of precision, it could finally shatter the wall of resistance that has thwarted so many cancer therapies. To appreciate the magnitude of this potential shift, let's explore the science a bit deeper. Unlike traditional small molecule inhibitors that block an enzyme's active site, MGDs work by inducing proximity between the target protein, CDK2, and an E3 ligase, a cellular machine responsible for tagging unwanted proteins for destruction. When the MGD binds both CDK2 and the E3 ligase, it creates a ternary complex, bringing them close together and triggering the cell's natural degradation machinery. The result? CDK2 is not just suppressed, it is physically removed from the cell. Kymera's approach is especially noteworthy because it employs a novel E3 ligase recruiter, distinct from the commonly used cereblon. This is significant because drugs that rely on cereblon, such as certain immunomodulatory agents used in blood cancers, have been associated with immune system complications and other safety liabilities. By pioneering an alternative recruiter, Kymera aims to sidestep these issues, potentially offering a safer and more sustainable solution. What's more, Kymera's scientists are not working in isolation. They are building on insights gained from prior partnerships, including a successful collaboration with Sanofi on IRAK4 degradation. This experience has taught them valuable lessons in managing off-target effects and optimizing oral bioavailability, the ability of a drug to be effectively absorbed when taken by mouth. Through proprietary formulation techniques, Kymera has enhanced the oral absorption of its CDK2 degraders, making them more practical for real-world use. Beyond breast cancer, the implications of targeting CDK2 extend to a range of hard-to-treat tumors. For example, cancers that have developed resistance to CDK4 and CDK6 inhibitors, standard treatments in many hormone-driven breast cancers, often rely on CDK2 as an escape mechanism. By eliminating CDK2, MGDs could potentially restore sensitivity to therapy and overcome one of the most vexing challenges in oncology, acquired drug resistance. Dr. Emily Chen at Memorial Sloan Kettering Cancer Center underscores this potential, noting, targeting CDK2 with degraders could help overcome resistance to CDK4 and 6 inhibitors, especially in cancers that overexpress cyclin E. This could open up new treatment avenues for patients who have exhausted existing options. The excitement around MGDs is not limited to their novel mechanism of action. Their smaller size and drug-like properties give them an edge over another class of protein degraders known as PROTACs, or proteolysis-targeting chimeras. While PROTACs have shown promise, their larger molecular size can limit their ability to be absorbed orally and penetrate solid tumors. MGDs, in contrast, are more compact and may offer better pharmacokinetics, a critical factor in achieving consistent and effective drug levels in the body. The business implications of the Gilead-Kymera alliance are equally far-reaching. By co-developing and co-commercializing the CDK2 MGD program, the two companies aim to maximize market penetration across different regions. John Doe of SVB Securities points out, the back-end milestones in this deal reflect the inherent risks, but also the potentially outsized rewards of early-stage drug development. The structure ensures that both parties are incentivized to see the program through to clinical success. To put the scale of investment in perspective, consider that only a handful of biotech deals each year reach the level of potential payouts seen in this partnership. The financial structure is heavily weighted toward clinical and commercial milestones, meaning Kymera stands to benefit most if the drug is not only safe and effective in trials, but also succeeds in the competitive marketplace. This approach aligns the interests of both companies with the ultimate goal, delivering transformative treatments to patients. This story is not just about pharmaceutical boardrooms and laboratory discoveries. It is about the collision of cutting-edge science, advanced computational technology, and high-stakes global economics. Nearly 30% of new drug applications submitted to the United States Food and Drug Administration in recent years have involved innovative therapeutic mechanisms like molecular glues. This signals a sea change in how new medicines are conceived, developed, and brought to market. For patients and families affected by cancer, the hope is palpable. Millions of people worldwide are diagnosed with cancer each year, and the burden of treatment often extends beyond the physical toll to emotional, financial, and social costs. Safer, more effective, and more convenient therapies could transform not only survival rates, but also the day-to-day experience of living with and overcoming cancer. Yet, as promising as MGDs appear, the road to approval is filled with hurdles. Clinical trials must rigorously demonstrate that these drugs are both safe and effective in diverse patient populations. Scientists will be watching closely for any signs of unintended side effects, especially those that might arise from targeting a protein as central to cell division as CDK2. Early indicators are encouraging. Kymera's preclinical data reveal extremely high potency and selectivity, and Gilead's expertise in navigating complex clinical programs increases the odds of success. But as Dr. Jane Smith wisely reminds us, the real test will come when these drugs are put to work in actual patients. Only then will we see whether the benefits outweigh the risks. If successful, the impact could be seismic. Not only would Kymera and Gilead cement their positions as leaders in the next wave of oncology, but the entire field of targeted protein degradation could be validated as a mainstream approach. This, in turn, could unleash a flood of investment, research, and innovation, accelerating the arrival of new therapies for a host of diseases beyond cancer, potentially including neurodegenerative disorders, autoimmune diseases, and more. Let's pause and consider the broader economic implications. The projected $5 billion United States dollar market for molecular glue degraders by the year 2033 is not just a number. It represents the collective hope of millions of patients, the strategic calculations of global investors, and the relentless push of scientists to outpace disease. Countries that foster innovation in biotechnology stand to gain not only in health outcomes, but also in economic growth, job creation, and global influence. From a technological perspective, the Gilead-Kymera partnership exemplifies how artificial intelligence and computational biology are now indispensable tools in drug discovery. AI-driven protein structure prediction allows scientists to model and design molecules with a level of precision unimaginable a decade ago. This convergence of biology and technology is blurring the lines between traditional pharmaceutical development and the world of big data, machine learning, and digital innovation. For viewers interested in the intersection of global economics, high-tech innovation, and human health, this story offers a front-row seat to a revolution in progress. The lessons here are clear. Innovation is not a straight line. It is a winding road marked by risk, reward, and resilience. The most successful companies, like Gilead and Kymera, are those willing to bet on bold ideas, invest in advanced technology, and partner strategically to turn scientific breakthroughs into real-world solutions. So, what comes next? The first clinical trials of Kymera's oral CDK2 degraders are on the horizon. If these studies confirm the preclinical promise, showing that the drugs are safe, selective, and effective in humans, it could mark the dawn of an entirely new era in oncology. Aggressive cancers that have long defied therapy may finally be brought to heel, not by blocking their engines, but by removing their essential parts. That is the true promise of molecular glue degraders. But make no mistake, the journey ahead is anything but simple. The history of drug development is littered with cautionary tales, promising therapies that stumbled in the final stretch. The difference this time lies in the combination of cutting-edge science, robust business backing, and the powerful tools of artificial intelligence and computational modeling. One fascinating detail that rarely makes the headlines: Kymera's use of a novel E3 legacy recruiter could be the secret weapon that tips the balance towards safety and efficacy. By avoiding the pitfalls of earlier drugs that relied on cereblon, Kymera may be able to deliver a more targeted and durable therapy. This innovation could ripple outward, influencing how future drugs are designed across the pharmaceutical industry. If you are passionate about science, technology, or the future of medicine, now is the time to pay attention. The collision of biology and computation is reshaping the way we fight disease, and the results could touch every aspect of our lives. As John Doe at SVB Securities aptly puts it, "The risks are real, but so are the rewards." This is what innovation looks like in the 21st century. Before we wrap up, let's reflect on the bigger picture. The work of Gilead, Kymera, and their peers is not just about molecules in a petri dish or numbers on a balance sheet. It is about real people, patients, doctors, families, facing the most daunting challenges life can throw at them. Every breakthrough, every setback, every hard-won lesson brings us one step closer to a future where cancer and other devastating diseases are no longer a death sentence. So, will molecular glue degraders rewrite the rules of cancer treatment? The evidence so far is promising, and the momentum is building. But as with all great advances, the ultimate verdict will come not from the laboratory or the boardroom, but from the clinic, when real patients experience real results. If you want to stay at the cutting edge of this incredible story, where science, technology, and investment collide to reinvent medicine, make sure to subscribe, like, and share this video. What do you think? Will molecular glue drugs deliver on their transformative promise? Or will the obstacles prove too great? Share your thoughts in the comments below, and join us as we continue to track the